It's on the bottom.
One more minute, 1 second. Just trying to get ready to go live. And okay. And I'm going to give you the controls, Amy. Hold on.
All right. What do you want to do here?
Do you want me to, let me see if I can give you,
Well, I will need to see slides though.
okay.
I'm gonna put that with the pasta, and the meat I'm gonna, and the meat I'm gonna fry with vegetables. The bean too.
Okay, go to the top.
Yeah.
You have to be at the other end.
It's like, yeah, you guys are covering the screen.
This
Hold
was
on.
not part of my show.
Oh, okay. I'm just trying to find how to go to the full
How
slides.
about, do I have control at all?
I gave, you're the co-host.
Well, I'm not able to do anything here.
Okay, let me, I'm sharing my screen. Do you want me to stop sharing and you share your screen?
How about, well, I don't have it on my computer here. I ran down quickly.
Okay.
How about if you just fix it so we start at the beginning and then open it up again and see if I can get it on my screen.
Okay.
Otherwise, I can pull it up from earlier. And good. Let's see if we can just. I'm not controlling it, Chris.
Okay, I'm not sure why I did give you the control, but I can you see it on your screen? I can advance the slides for you.
Sure. There's going to be a lot of next.
Okay.
Okay. We'll try it so we don't tie anybody up. All right.
Okay.
So hi. I'm Amy Fowler. I think I know a lot of you on the call. I'm the chair of dermatology and professor of pediatrics at Northwestern University and the Lurie Children's Hospital, and have long been involved in FIRST. I think this is something like my 27th year of participation in FIRST, and always been a pleasure to talk about all things, including research. So let's go to the next one, Chris. So we are lucky. We're seeing more and more finally on research in ichthyosis. And research is very important. We can increase our understanding of these genetic changes and how they lead to the scaling and redness and other features. We can develop the best measures for severity, both observed and as experienced by someone with ichthyosis. And these are incredibly important if there's going to be research to develop new medications. We're going to be finding markers here that help us to predict the course, the risk of getting other medical issues, and best treatment approaches, including on a personal basis, this personalized medicine we keep hearing about. And very importantly, we're going to discover new treatments. You know, what's happening? Thank you. You know, it would really be great if I could control this. Maybe I should just pull it up on my computer. If everybody will excuse me, I'm going to go out for a minute, pull it up, and we'll go from there.
No problem. We've got a couple of staffers here, and we jumped right in, so we can introduce ourselves and say hello.
That would be great. Why don't you do that while I get up my version, and then I'll see what I can do here.
No problem. So I will warn everybody, Dr. Fowler is brilliant, and these slides are quite dense. We are thrilled to offer this on playback later. So this will be available to you later. So if you can't get everything down the first time, don't worry. And if you're watching this in playback later and you still have questions, you can always reach out to the office and we can try to clarify for you. So don't worry about it. Don't worry about it right now if you are overwhelmed today. My name is Denise Goss. I'm on staff at FIRST. Prior to being on staff, I was on the board for 4.5 years. I myself have CIE. I'm one of 4 affected children, and this Ichthyosis Awareness Month has been a fantastic opportunity for all of our stories and all of our families to come together and share and hear and listen. We've had so many wonderful things happening all month long. We've had several people running fundraisers. We've had members sharing their stories every Monday, and we're wrapping it up. It's been a really fantastic entire month full of virtual events as we have learned how to fill in for all of the in-person programming that we're missing so dearly. So coming up next week, we've got 2 more wonderful events. We have a Member Monday with Hunter Steinitz, who is just an incredible young adult with Harlequin ichthyosis. She'll be sharing her story. She recently graduated, or is about to graduate, I think, from theological school, and she's also got a background in acting. So we look forward to hearing from her Monday night at 8 p.m. Eastern. Is it 8 p.m. Eastern, Chris? I always get the times wrong when I start talking live. Okay, I think it's 8 Eastern. And then on the last day of the month, at the conclusion of the Ichthyosis Awareness Month, we have another Release the Butterfly encore concert event. So earlier this month, there was an incredible ensemble cast performance of Release the Butterfly as a fundraiser, and we are so excited to bring them back. And so speaking of fundraising, we are also fundraising this month. Of course, our campaign for the month is I Am 2020. In addition to it being the year, it is our goal. It is our goal for 20% of our members to donate this month. $20 is the recommended amount to chip in. If you can give 5, it means so much to us. Historically, about 5% of our membership donates. And as you can imagine, as a small nonprofit with a very small, very small, very rare skin condition here, every dollar counts. So we are right at about 4% participation right now. So we're trying to really drive it home. So over the next week or so, if you haven't already donated, please consider chipping in whatever you can, just so that we can really turn up the participation numbers and show that we really are all involved, we're all invested, and we're all actively supporting everyone else in this ichthyosis family. So that's it for me. I'm going to turn it over to you, Chris, and Lisa. I know that you guys are both, I don't see Lisa at the moment, but Chris, would you like to introduce yourself?
Sure, I'm Chris Boynton. I have just recently joined the staff of FIRST just on Monday, so I'm on day 4, learning a lot about ichthyosis. I know I've met a lot of great volunteers, and I'm very, very excited about working with all of you and meeting you all very soon.
Awesome. Chris Wassel? Oh, you're on mute, Chris.
How about I go?
Okay.
You're still on mute, Chris. So I'm Lisa Bruning. I've been with FIRST for 12 years. The last 5 years have been full-time. I'm the director of operations. I manage the website, and I handle, when the phones ring, either Chris answers it or I answer it. So we're always here to help you with whatever you need. And no, not quite.
Can you hear me? For
Yes.
some reason it keeps muting it again. I'm Chris Wassel. I'm the Community Engagement Director at FIRST, and I'm thrilled to be here. And let's kick it back off to Amy. I think she's all set.
Yeah, I can't see any of you right now, but hopefully you can see my slides and they're ready to roll. So I'm gonna just take off where I left off before. So we're talking about the excitement that there's more and more research on ichthyosis in a variety of different ways, and ultimately down to the line, discover new treatment approaches that are going to be maximally safe and effective. And you, if you haven't gotten involved in research, should become involved, not just help you, but also to help others in the next generations with ichthyosis. It's so important. So where are we with ichthyosis research? Well, first, I can't not start with the registry. I hope that everybody who's watching today with ichthyosis or knows somebody with ichthyosis encourages them to get involved in the registry. It's been so important for us to understand the genetics of ichthyosis, and is equally important for us in doing other kinds of research. It allows you to be part of a community that's contributing. We are collecting information, for example, about itch, quality of life. I'm going to talk a little bit about that. It's free to find your gene. If you don't know what is causing your ichthyosis, it's really important to get this done. It's being done in a research lab. Keith Cho, one of our really important advisors here at First, is doing this research, and he lets your doctor know, who will let you know then what gene is involved. But he can't tell you the exact genetic change, but for very little money, in addition to that, we can make that work for you too. But very importantly too, is to sign up through the registry to be contacted about research. And of course, First is another wonderful resource to tell you about potential future research studies, because you should get involved. Now, one of the things that came out of this is about mental health. So there are all these different types of questionnaires that you're answering as part of this. And a young student at Yale took this information and just published this in one of our major dermatology journals. And it tells us about ichthyosis. So there were 181 individuals, a mix of adults and children 8 years and older from the registry. So she was able to look at the quality of life and anxiety and depression, and found out that 95% of adults and 85% of children experienced impaired quality of life, that 34% of adults and 30% of children screened positive for depression, and that 27% of adults and 38% of children screened positive for anxiety. And this was all pre-COVID. So these are higher odds than of having depression or anxiety with more impaired quality of life. Why is this important? Why am I telling you about this? Because we need to have these kinds of studies that show the high, what's called burden of disease caused by ichthyosis. So many people over the years have dismissed all kinds of skin problems as, oh, it just involves the skin. It's not life-threatening. What does it mean? And showing this kind of research is very important because, first of all, high burden of disease helps the FDA to make a decision about fast-tracking new medications for testing. And there's greater interest from the pharmaceutical industry in drug discovery for ichthyosis if they can then show burden of disease and get insurance companies to cover it. Now, part of the burden of ichthyosis is also itch. And this is a study that just came out with almost 100 adolescents and adults, different kinds of ichthyosis, where they were looking at the frequency, the duration, the severity, and the impacts of itch. 93%, almost everybody, experienced itch. And the average was 4 out of 10. So not the most severe, but still itch. 63% found it was often or always present, although mostly less than 30 minutes in duration. But they were able to look at subtypes as well. And with Netherton syndrome, certainly one of our very important forms of ichthyosis, it tended to be the worst itch and the longest lasting. And the least was in recessive X-linked ichthyosis, one of the most common forms of ichthyosis. Triggers were delineated: change in weather, hot environment, stressful situations, and consequences were also shown. All those scratch marks, hard to fall asleep, bad mood, loss of concentration. Now, this was a study that was using a scale called the Leuven Itch Scale, which was only validated, meaning was only shown to be effective for adults. They extended it to those more than 12 years of age, but we can't just use scales in adults that aren't applicable for children. I'm happy to report that there's a new itch scale specifically for children that First, and hopefully many of you were involved in helping to make. So this is a tool that was developed with support from the National Institutes of Health that measures severity, quality of the itch, the impact of the itch in children. It's not adults, it's children specific, and can be used for children as young as 8 years for self-reporting, but also parent proxy for 5 years and above. And this has gone through extensive what's called validation to show that it is good as a measure, and it will be used moving forward by the drug industry for this. Now, this study was just completed for validating in more than 250 children with eczema, but before that stage, there was a whole stage with calibration where the questionnaire was sent out to more than 600 children, and many of those were with ichthyosis through the Foundation for Ichthyosis-Related Skin Types. So really delighted. This was an example of how you can help. Now, this is undergoing FDA review now, and it's in testing for children with other itchy skin problems, including ichthyosis. So why is this important? Well, itch is another huge burden of ichthyosis, as I just showed you, has a significant impact on quality of life, including on sleep, including on mental health. And when the pharmaceutical industry is trying to develop new therapies that are going to improve inflammation and as a result, itch, or drugs that can directly reduce itch, having a valid measure like this is important to show impact. So what are some of the other measure studies that are coming down the pike? Well, it's very important to have a good validated measure to talk about how severe ichthyosis is. And many of us have been developing and using measures because there hasn't really been a good one. So I'm happy to report that Keith Choda and I teamed together with a student to really move forward what we're calling the Ichthyosis Severity Score. And it's going to be assessing redness and scaling in the entire body and creating a composite score that's similar to what is now available and validated for common skin problems like eczema and psoriasis. This is looking very good in validation testing, and it will soon be available for medication trials. I also want to tell you that the Pediatric Dermatology Research Alliance, which has more than 200 doctors who are interested in doing research involving children, chose as its big study one that looks at stigma, depression, anxiety, and quality of life specifically in children who have visible skin disorders. Now that is well on its way towards having 3,500 children across the country, including many with ichthyosis, and it had to be stopped. It was stopped because we've not been seeing anybody in person, and this is a study that has to be done in person. And quite frankly, we're not exactly in times that don't in themselves potentially cause anxiety and alter quality of life. So that study's on a hiatus. It's on a break until September, when we're hoping people might be back in school, certainly back in our clinics. But if you have a child who has ichthyosis, you should reach out to your doctor if you have a pediatric dermatologist and ask if they're part of this study, because there are 30-some centers that are involved, and we'd like to get as many ichthyosis patients involved as possible. This was a study that we were going to do at this year's annual meeting, but unfortunately it couldn't happen. Now we are going to be able to ask your help in doing some questionnaire studies. And pretty soon, FIRST is going to send out a posting that will tell you that if it's your age appropriate, please help us out. And I'm going to just mention these. So for 8 to 17-year-olds and a parent, we want to understand how often children and teens with ichthyosis experience bullying, including cyberbullying. And this study's almost finished going through our institutional review board. Quality of life in ichthyosis. We've used in studies quality of life measures that are out there. And what we found is that people found great change in their quality of life, but it wasn't reflected in the scoring. And that tells us we need a new tool. So we're trying to develop questions that address the most meaningful life changes. For example, burden for household activities. So this will be going out for any of you who are adults. And then just understanding what your understanding is of ichthyosis and the treatments that you use. This is so important for us because it's really a critical part of adhering to therapies and getting the best benefits. So this will be actually for adolescents and parents to teach us how well you understand these aspects. Now, I also want to mention another measure, which I think is really interesting. So as you know, all forms of ichthyosis share one thing that's very important, and that is having a very poor skin barrier. Having a poor skin barrier means more water loss. That's why your skin looks so dry. And easier penetration of drugs and environment because you have a poor barrier. And that contributes to inflammation and scaling as a response. Transepidermal water loss is a term that means that you are constantly losing water through your skin. We all do to certain levels, but there's much more loss in ichthyosis. And this transepidermal water loss, or TOOL, can be measured as a surrogate for barrier function. So this is a study that's just in press with 122 children and adults with what we call the orphan forms of ichthyosis. So not a common form like Ichthyosis vulgaris, or even a somewhat less common form like recessive X-linked ichthyosis, but what are called orphan forms, where there are less than, I think it's 200,000 individuals who have this in the US. So we measured the transepidermal water loss, and we also correlated with the severity of the redness and scaling. And what we found is that the mean transepidermal water loss was about 2.5-fold that of our control patients, highly significant. And by the way, if you don't know how to look at p-values, which show you if something is significant, less than 0.05 or 0.05 is considered significant. So this is highly significant. We found it to be the highest in Netherton syndrome, intermediate in those patients who had congenital ichthyosiform erythroderma, ichthyosis with confetti, erythrokeratodermas, lamellar ichthyosis, and lowest, although still elevated, for epidermolytic ichthyosis, and we had one patient with a rare form called trichothiodystrophy. We found that there was an inverse correlation with age. So that means that there was the greatest transepidermal water loss in the younger individuals, suggesting that as you get older, you are able to compensate somewhat, perhaps through thickening of scale, and that the one disorder in which there was the greatest correlation, and it was an inverse one, was transepidermal water loss. And that was with the scaling severity measure. So those who have lamellar ichthyosis tend to have a lot more scaling as a response to having that barrier abnormality than the other forms of ichthyosis. So this was actually very interesting. But transepidermal water loss is something that is interesting, but it's measured with a very fancy and expensive machine. So the gold standard for measuring is something called the Aquaflux. Like I said, it's a big clunky machine. You have to wait about 20 minutes and then you can do the serial measurements. We always do 3 to make sure that we're not just relying on a single one. It takes time. And who has these? We can't easily do it in the office. But more recently, there's been a pocket-sized affordable device that's measuring tool that's come out on the market. And so we did a study, and many of you were involved because we did it at the last first meeting where we sat there and we used these 2 devices to measure. And you can, if any of you did that, you might remember that we used the big clunky one or we used the small one, and we alternated person by person. Here are the results. What we found is that the small device and that fancy Aquaflux one were strongly correlated. The R up there tells you the correlation, and 0.743. It's a very strong correlation, and there's that p-value. I told you 0.05 is significant. Here we're 0.001, so very significant. And when we looked at what's called test reliability, meaning if you did it and then you came back and did it again, it would be the same. So when we did triplicate measurements, we were able to look at that. And you can see that this is true across the board for a variety of different forms of ichthyosis: lamellar ichthyosis, epidermolytic ichthyosis, congenital ichthyosis from erythroderma, Netherton syndrome, and a variety of others in comparison with the controls. And you can see the controls are all those black dots at the bottom. And you have little pictures of what we're talking about here for those of you who weren't part of that. And you can see this very tiny pocket-sized handheld device, which is wonderful too, because the minute you take it out of the pocket, you can measure it without having to wait. So very exciting, and means it's that much easier to do studies where we measure barrier and the impact on the barrier of using different agents, whether that's a moisturizer or whether that's a prescription medicine. And by the way, the Aquaflux costs $22,000. The little one costs $250. All right, so what about ongoing clinical studies? Well, retinoids. Retinoids have been around for a long time. They're our favorite drug to use orally or topically if we can get for our patients. They're especially useful for lamellar ichthyosis and recessive X-linked ichthyosis. They're not well tolerated by Netherton syndrome and variably by some of the other forms like CIE and epidermolytic ichthyosis. But there are two new topical retinoids that are in development, and these are phase 2 studies that are ongoing. I want to tell you about both of them. One of them is a topical form of isotretinoin. Now, isotretinoin is this wonder drug that we've had out now since 1982 for acne, and it works great for that. And we also use it for ichthyosis, but it's never been available in a topical formulation. And the other is a different retinoid called trifarotene. And you can see both of these studies. I'm talking about the names for the studies. They're both on the first website. And if you're not familiar with clinicaltrials.gov and you're interested more in what's going on in trials, just go into that website, very easy. And each of these have a special number because if you're going to do a study, you are required to register it in clinicaltrials.gov. Now, I'm going to just talk about a few terms that I'm going to be using coming up as I talk about these. One is randomized. This means that each participant in a study is assigned by chance. And it doesn't matter what the previous one had. It's like flipping a coin. You may have a 50-50 chance, or you have a 1/3 of being this, 1/3 of being that, 1/3 of getting that. The next definition is double blind. Double blind means neither the doctor nor you know the assigned treatment, and everything you get would look the same. Vehicle controlled is for topical studies. It means one of the treatment groups is the active medicine and the other just has the base, the vehicle that the active medicine is coming in. And that's for topical trials. If there were an oral trial, you'd be taking something that's a pill that looks like the pill. If it's an injectable trial, you'd be getting an injection with just salt water in it or something like that. Now, the two here we're talking about, both are having 12-week randomized double-blind trials, but they're a little bit different. The topical isotretinoin has already had an earlier study suggesting the best dose and showing some safety in 19 adolescents and adults compared to that vehicle, whereas the topical trifarotene is doing that study now and beyond. So the topical isotretinoin is a possibility of being randomized to either the vehicle or the active for 45 participants 9 years of age and older with lamellar ichthyosis or recessive X-linked ichthyosis involving at least 10% of the body surface. And there are many international sites. Check it out if you're interested. You'll see just who's doing this if you go online through FIRST or through clinicaltrials.gov. Same thing with the topical trifarotene. Here, though, there are 3 arms. So you've got a 2/3 chance of getting the active medicine, and it's 120 adults and adolescents with just lamellar ichthyosis. So you might get one of 2 doses of the medicine or the vehicle, and you have an option to go into an open label phase after this for another 12 weeks with application, again, up to large areas of the body, international sites available. So check these both out if you're interested. Now, I want to talk a little bit about pathogenesis-based therapy, because this is really cool. We're learning about all the genetic changes that underlie these disorders, but how does that translate into therapy unless we're talking about gene therapy? I'm just going to show you some examples of this. This is work that we published almost 10 years ago, knowing the cause of a rare form of ichthyosis called child syndrome. You can see the red scaly skin here in this picture, and that it was part of the chemical pathway of how cholesterol is made. Lots of stuff here. I'm not going to give you a quiz later, but what's circled in red is the gene and where it plays a role in our making cholesterol. We all need cholesterol for many, many things, including the health of our skin. Well, it turns out that these individuals were having a block in the progression of this pathway, so they were accumulating all kinds of toxic in-betweens that's causing this problem. So we all know that at the top of the cholesterol pathway is something that is a rate limiting step in making cholesterol that is blocked by statins. Statins are all over the place. So many people are using them now because they have high levels of cholesterol. Well, we can do this same thing in someone who has a disorder of the cholesterol pathway, because we can block that by using a topical statin. We can add back the cholesterol that's missing so the skin stays healthy. And look at what happened in this patient. So wonderful response over a very quick period of time. Just in a 6-week period, we can already see good results from this. So this is a concept of how, knowing how things get to the redness and scaling, we might be able to block something. Now, what's hot off the press just this year is there's another scaling disorder that's part of our family of related skin diseases in FIRST, that's called porokeratosis. And it's been discovered that the genes that are altered in porokeratosis tend to again be part of the same pathway. Now, these are conditions where there are just stripes of involvement, because it's probably not even compatible with living if your entire body had this, but we can see stripes of involvement in porokeratosis. So when we learned that it's the same cholesterol pathway, we said, aha, wonder if we can do the same thing. So once again, we block the statin, we use a 2% statin, we use a 2% cholesterol, just like I showed you before, and you can see some of the results. So when this was put on, we can see someone with extensive porokeratosis on the left, marked reduction, can barely see it anymore. We can see a marked reduction after just 12 weeks with the one on the hand here. And then somebody who has it all over the legs, this is an interesting genetic condition in which the sun and age bring these out, particularly on the legs, and you can see the dramatic improvement after even just 4 weeks in this first picture. So a new therapy just from understanding how things work. Well, can we think about other ways to look at what's being produced in ichthyosis and try to prevent the redness and scaling? So this is a study, I think some of you saw me present a little of this before, which we looked at a lot of different types of ichthyosis, all listed here, but some of the more common ones involved in FIRST. And we've published several studies with more coming, where we looked at the skin of individuals with ichthyosis, and we've looked at the blood of individuals with ichthyosis. And this was a study that we actually started a few family meetings ago, and we also certainly were continuing this at our last meeting. So I hope some of you were part of this. And as you can see, we looked at selected genes originally in skin to see what is increased, what there's too much of, what there's too little of, how it changes. We were then able to do big data, which means we could take that same material and look at more than 47,000 genes. And we've also looked at what's going on in the blood. And here you can see, this is the kind of thing we can see. We take our 4 major types here: Netherton syndrome, lamellar ichthyosis, CIE, epidermolytic ichthyosis, and we can do what's called the Venn diagram. And with all of these about 47,000 genes to look at, we can see that there are some that overlap among all the different forms. That's what you're seeing in the middle here, this 113 and 19. And then there are plenty of genes that also are not shared and are what make each of these different forms of ichthyosis unique in how we see them. Well, I hope we can study all of those someday because we've got all this data. But right now we wanted to look and see what's shared. Can we take that information and try to move it forward towards some type of therapy? And here, as you can see again, towards the middle, 113 shared ones that are going up, red means there's too much, and 19 that are going down, meaning there's too little. Get rid of that. And we can then look and see when we put those all together and compare them with 2 very common skin problems in which we really know what's happening genetically in those in terms of what there's too much of, too little of. And we can see that there's a huge overlap with psoriasis, a common inflammatory skin disease, 2 to 3% of the population, and really not that much overlap, at least exclusively with eczema or atopic dermatitis. And then there are some, of course, that don't overlap. So that was very interesting, and we were able to look at what's called a heat map, and that shows an array of all these different genes. You don't have to know what they were. You just have to see that 2 of those columns are red. That means a lot of increase with PSO, psoriasis, and ICH, ichthyosis, and we're not seeing them in the normal skin at the left that's all blue, and we're not seeing it much in eczema or AD there. Now, what can we do with this information? That's so interesting because we ask, is this kind of a pattern? And what we're seeing there especially are certain immune markers that are being raised. It's like certain arm of the immune system is being activated here. Is this pattern, because we know these are increased when we're fighting off bacteria or fungus and other organisms that may be at the skin surface. We know ichthyosis has a poor barrier. This is just natural. These are just going up as a result of that. But there's also the chance that it's actually this skewing of the immune system that drives inflammation and scaling, as for psoriasis, and then could targeted therapy treat ichthyosis. So I'm going to show you a few examples of case reports and series that suggest we may be onto something here. This is a little girl who has a form of ichthyosis. It comes along with heart issues, and that's why she's had that surgery. You can see a scar down her chest, but she's got a lot of scaling. Her whole body's so red. She's so itchy that she's putting on topical steroids twice a day, and her barrier is terrible. So she's puffy as if she's taking steroids by mouth. We were able to look at her, and here you can see some graphs that I'm just going to walk you through, because what's important to recognize, it's not really what's on here, but what the punchline is. So if you look at the graphs on top, what you see is that this little girl, who's that red dot on the left, has a lot of something called interleukin 17 or IL-17, just like in psoriasis. And she's got other things that are also increased, like in psoriasis, as shown, for example, in the red. Now, she also had what's shown in the purple, and that helped us make a decision on what medicine to put her on. Now, if you look down below, you see all these bright colors, these kind of interesting graphs. These are called flow studies. So we can take her blood, we can run it through a machine that separates things out and shows how many of her cells in the blood have on their surface and then shed into the bloodstream to be detectable elsewhere, certain of these immune markers. And I'm showing you increases in something called interleukin 22. And my screen's cut off at the bottom. I don't know whether you can see it, but increases going the other direction at the bottom in the same interleukin 17 that I showed in green at the top. And just take my word for it, that's what it's showing. So not just in her skin, but also in the blood. She in particular had an increase in these 2 immune chemicals that really we know can drive inflammation. So this girl and a boy with a similar genetic change were treated with a drug called ustekinumab, which we know inhibits these pathways that we identified from the research. And if you look carefully, you can see that they look almost clear. These were life-changing for them, and 2 years down the road, they're still doing very well. I'll tell you, in the case of this little girl that I talked about a minute ago, 2 weeks into it, she was able to stop the topical steroids because her itch went away. And she suddenly looked like she lost all this weight because she wasn't puffy anymore. And similarly with this boy, he's doing extremely well. And by the way, has not gone on to develop the heart disease. Now there's another syndrome that's called SAM syndrome. You can see it defined at the top here. And this is an 18-month-old who similarly shows what looks like ichthyosis, redness, scaling with this SAM syndrome. This was a case from Spain. And because of knowing about the work that we did, they were able to look and see that this child also had this activation with 67-fold more of that interleukin 17 positive cells in the blood, 38% more of an increase in the protein itself. This is after 35 weeks. of monthly secukinumab, which is a drug by injection that specifically lowers interleukin-17. And in fact, this child was shown to have fewer of those cells expressing the interleukin-17. That's shown here on the top with the reduction in those bars over time. And then what you see at the bottom, which is really important, as part of this syndrome, these children were just not gaining weight. They had metabolic wasting. The minute this child was put on the drug, and I have a child I'm treating with this exact same thing too, very soon thereafter, the weight almost went up in a vertical line. Very rapid increase after that to the 50th percentile, and that's what's happened with my child too, and a marked reduction in infections as well. Now, there are also some reports of benefit in particularly those forms of ichthyosis that are redder, Netherton syndrome and CIE in particular. I'm showing some of those that have been published here. On the left, you see a 9-year-old who got that ustekinumab that I talked about when I showed you the example with the cardiac issues. Getting it every 8 weeks versus every 12 weeks, he did the best with that. And on the right, we have a 20-year-old with a form of CIE who has monthly secukinumab and is doing quite well. Well, does it work for everybody? No, it certainly doesn't. And I know from personal experience, because we did a secukinumab study in adults and our results were less exciting. We had 5 who had life-changing results, wanted to continue, but most didn't. Nobody with lamellar ichthyosis, unfortunately, did any better, and others as well. We couldn't even just sort it out, but we're doing research now to try to understand why some respond and others do not. And I don't think that that closes the door, because we really need to understand why some people do well and others don't. And I'm really excited about 2 trials that are coming up. One will just be in Chicago, where we're taking ustekinumab and we're going to treat children as young as 6 years of age. The Food and Drug Administration said we could do that. There's been good testing now for psoriasis down to those ages. We know it's very safe. And we're able to do every 8-week dosing. So I'm going to be able to come back, I hope, in the future and tell you about how that does. And then there's also going to start a multi-center trial of a different inhibitor. Maybe it's more potent, or maybe it's just going to work in a different way for adults with the inflammatory forms of ichthyosis. And there'll be several centers across the country on both coasts and in Chicago to be able to be part of that with a travel budget for doing it. And many of these studies, including the 2 I told you about retinoids early on, have travel budgets so they can get you other places. Of course, once this pandemic is over, we're going to want to travel again, but not right now. But in the future, when these are started, hopefully you'll be able to take advantage of that as well. I want to tell you about some very exciting gene therapy research. This was presented recently at the Society for Investigative Dermatology meeting. This is a phase 1, so the very first, very beginning exploratory study of gene therapy for lamellar ichthyosis. And what's exciting is this is a topically applied gene therapy. So this is what we call an open label, meaning everybody got the real thing. Intrasubject, meaning that in the same individual, they had both the vehicle, the placebo control, or they had the active medicine with the actual gene that's missing in it. There have only been 3 treated so far, and these were done in Philadelphia, but we already started a study with 3 more in Chicago. This is going to start with adults, and they have to be adults with lamellar ichthyosis who've proven to have mutations, to variants in transglutaminase 1, and therefore are deficient in it. That's the gene that causes lamellar ichthyosis. They also have to be moderate or severe based on the assessment of severity. And this is to demonstrate some safety, decide that it works, and really decide how to move forward. Now, I will tell you that before any trials go to humans, there are preclinical trials, and they're done in cells, and they're done in mice, and sometimes in other animals as well. So when this was added to cells in culture from people who had ichthyosis, the cells started making the protein, and it was functional protein. It wasn't just made, it worked. When this was applied to mouse skin that had a bad barrier through what's called tape stripping, it's basically taking something like Scotch tape after you shave the mouse and you just take off some of the outer layers, so a poor barrier. And putting it on topically, the protein was found to get in, to be at the right place, and to be functional. And all of these studies in the mice showed that it was in the skin, not inside, and that the mice were healthy. So 3 patients were included. I'm just showing you their skin here. No question, this is lamellar ichthyosis, and these were severe, moderate in severity. So how does this gene therapy work? Well, it's called KB105. It's formulated into a gel for direct application to the skin. And the companies put multiple copies of the gene that's missing, the transglutaminase 1, into what's called a vector that is a virus vector. But it's very safe because the virus doesn't get inserted into our genetic material and also is not able to multiply. The system's very efficient at introducing the gene, and its manufacturing is expected to be easy. The vector can easily and be frequently applied to large areas. It's topical. And in the future, it's going to be able to be administered by a healthcare worker or even a caregiver at home after the component parts are put together. To my knowledge, it's the only lamellar ichthyosis correcting gene therapy in clinical development right now. Now, lamellar ichthyosis was not the first problem that's been broached with this, so we have some good information. This has been used for another genetic skin disorder that's a blistering disorder called epidermolysis bullosa, and it showed good introduction of the gene. This was a collagen gene that's missing in that disorder. No evidence of exposure outside of the skin, no immune responses, those would be unwanted, no evidence of the virus, and no evidence of any serious side effects from the drug. So this is an example of how this moved forward. These were 20 centimeter squared circles that were put in similar areas of skin. You can see here's an example where we have a placebo site and we have a site for active that are separated. And just in brief summary of these results, this introduced the DNA that was supposed to be getting in, and it produced the correct protein at the right place in skin, and it was active. This occurred just by putting it on. The company tried to see if it made a difference to do what's called microneedling, which is a physical way to get it to penetrate the skin barrier. It didn't make any difference, particularly as it was reapplied. And it was sustained and actually even increased with repeated administration in terms of what we're seeing with the protein in the skin. So I'm showing you some examples here. If I were to show you somebody's skin from lamellar ichthyosis, you would see no green here. So this green is specifically recognizing the protein that's different. Here we have it on day 3, 1 treatment, and you can see it's already starting to sit there. And I will tell you that that outer area of the skin is where it's supposed to be. And if you look over in the picture marked day 29, this patient was serially retreated, and this is 2 days after the last retreatment. And I'm going to tell you that that's beautiful demonstration there of the transglutaminase 1 protein in the skin. Now, the optimal dosing seems to be weekly, and I'd say it's a little too early to know about if it looks better in those little spots because they're such small areas, but it looks promising. There seems to be less reverting back to the scaling in the treating areas, but of course, it'll take some time to look for that. These early studies are largely safety and proof of concept. It can get in. There was no evidence of a safety issue here, no side effects, no evidence of an unwanted immune response to the virus, no new antibody produced against the transglutaminase 1 protein or against the virus itself. There was no evidence of shedding either in the blood, in the skin, in the urine. So what's coming with this particular exciting agent? Well, the exploratory trial is going to be continuing on. Excited about that. We're going to be doing that once weekly dosing over larger body surface areas, and then moving to pediatric patients, larger numbers, and getting approval for home application will be very important for that moving on. I want to finally end with some pathogenesis-based therapies for Netherton syndrome that are coming up. So Netherton syndrome is all related to changes in one gene called SPINK5. And what happens is that you don't produce anything called lectin. It's a protein that prevents the breakdown, particularly of proteins, and it allows an increase of kallikreins particularly kallikreins 5 and 7, which are involved in breaking down certain proteins. So as a result, when these kallikreins go up, we see breaking down of proteins that make up the skin barrier and also driving skin inflammation. The new therapies in development are, first of all, targeting these kallikreins. So they can be targeted with agents that are called small molecules that can be put into topical formulation and put on the skin. Or there is also people who are thinking about using gene therapy topically to suppress the expression of kallikrein. So this is all out there and moving towards trials. And then there are also gene therapy approaches that can add back the normal SPINK5. So just like what I showed you with lamellar ichthyosis. In fact, that company is moving forward in time with being able to add back the SPINK5 gene and hopefully then reverse the deficiency. But there's also another way that's being looked at in Europe, and this is a situation in which this is cellular gene therapy, in which you're introducing the gene for the SPINK5 into the Netherton skin cells. Now, this has actually been done with great success for, again, that other genetic disorder that blisters called epidermolysis bullosa. There are a lot of studies going on right now, particularly at Stanford with grafting, but this was a study that was done and published in a very, excellent journal. It was just a terrific, exciting development in which a boy with epidermolysis bullosa who was 7 years of age had most of the body missing skin. And you can see in red and green all the areas that were blistered or just lost. And they did a single little biopsy, 4 millimeters, from a non-blistered area in his groin area. If you see on the front part of him, there are some areas that weren't blistered. They grew that out massively. They introduced the missing gene and then were able to grow it out further. They expanded greatly the skin cells, and they tried to make sure that they were using what are called stem cells, the cells that have the capability of growing and growing and growing long-term. And they made a lot of these and they checked it carefully, and then they transplanted this large sheets of skin to cover all of these blistered areas. You can see the boy after that transplantation. And I'm told, and in the publication, it showed that the missing protein was now there. Just like I showed you before, where there was green, where there wasn't before. Now this boy is making his protein that was missing. And in talking to the investigator on this study, a few years later, he said this boy still has not a single blister in the transplanted areas almost 5 years later. So this is moving forward in early phase trials in Europe for Netherton syndrome, but this time putting in the SPINK5 gene. So in conclusion, growing research to help us understand ichthyosis, develop and test new measures, and treat in different ways. Think about getting involved. Fill out those questionnaires if they come to you. Take a minute to do it. Look at the first website to find out what trials are available for your form of ichthyosis, or go to clinicaltrials.gov, and even think about traveling in the future to help out with this kind of research. We may see a little bit different research in the future. There are some companies out there that actually, at least in between visits, go to your home and take pictures and can draw blood or do whatever is necessary. I think we're going to see those more in the future, especially starting up during this time of the pandemic. So that may be another option too to decrease travel. Overall, final conclusion, the future looks bright for drug and gene therapy for ichthyosis. I'm very excited. I hope you are too. And with that, I'm going to stop. Thank you for your attention and happy to take questions. I hope you have some. Okay. And I'm back.
I have a
We do
question.
have, okay, there are plenty of questions, but go ahead.
Chat room. Oh my gosh.
I was happy to hear a lot about lamellar ichthyosis. Our daughter has lamellar ichthyosis. My question would be on all of these studies or trials that are going on that are topically or treating the skin, would any of those also, if they had positive results, would that also fix the inner body temperature issues with overheating and getting cold super easy?
Okay, so the issues with overheating largely relate to the fact that sweating is an issue. And I think it's a really good question because some people, for example, who respond very well to the oral retinoids are able to start sweating again, or sometimes we've seen as people get older, they have the capacity to sweat again. But sometimes we seem to be treating very effectively and there's still a problem with sweating. And that suggested to me, I wonder if there's a developmental issue in the developing sweat glands or something from all of the extra scale. So I think the answer is we don't really know. Theoretically, if the skin goes back to normal or near normal, it should fix all that if it's a problem that's based in skin, which we think it is. But if in fact there is an element that reflects having some change early on in life, or even with that collodion membrane when things are developing inside, that might suggest that it couldn't be reversible. So I hope so. The topical medicine is not making the difference versus something that gets inside, because I think that is a skin problem. It's just the question of, is that reversible, as opposed to the scaling, which absolutely should be.
Hello, Dr. Power.
Yeah.
This is Sam Simmons. How are you? Yeah, thank you for your presentation and information. Really appreciate it. Question regarding the gene therapy for lamellar. I recognize that it's in the early stages in the trial. How familiar are you with the drug development process and the potential timeline on when that might be available on the market?
Well, I think that's it's fast tracked. It's going quickly, but obviously you have to get through the early studies. So it's still in phase 1, but it will go through other phases. Sometimes there's an ability to combine phase 2, phase 3. I would say it'll be a few years because you really have to have that safety, but it's an opportunity to get involved in trials because those trials will be in several centers, I'm sure, in the future if the early trials look good. Now, I see a whole bunch of questions in chat room. I don't know if people want to just pipe up their questions or if you want me to just run through some of those. Chris, tell me how you'd like to run this.
You know, Amy, if you go through them and you see something you want to answer, just go right ahead.
Okay, I will zip up to the top here. Interesting. Okay, polyarthritis and ichthyosis. Really important question here because we think that that's a really important association that's been very much understudied. I know many people that I know with ichthyosis, when they get to early adult years even, start to have issues with arthritis. And several of my young children with harlequin ichthyosis have had issues with arthritis as well. And one of the exciting discoveries was that we found in the blood, and I didn't have time to go into this, we found in the blood that and I actually did mention it for that one girl with the heart. We found this across the board in ichthyosis as well, that in the blood, the cells were expressing more of this interleukin 17, interleukin 22 in particular. And that always raises the questions, is this a systemic problem? And I think it is. Psoriasis is a systemic problem. There can be an association with arthritis. There can be an association as well with things like cardiovascular and metabolic issues. So we're keeping an eye open. I haven't seen evidence, at least in our small study, for any metabolic issues, cardiovascular. However, we do know that there seems to be more arthritis. So one of the values of these injectable biologics for psoriasis is that they can help with arthritis as well. And particularly the interleukin 17 blockers, I think, do very well with that. Stelara is okay. It's not as good for joints as some of the more IL-17 specific ones or some new ones that have come out for adults that are more specific to something called IL-23. So there are a bunch of them out there. Rheumatologists know all about them if there's something with arthritis going on. Unspecified liver complications. The biologics really don't seem to have problems with the liver. So that's one of the reasons why they might be really a good try. I really think that the younger somebody is, the better they respond. And there's an article coming out this month in one of our big journals with several with Nethertons who responded to one of these, and I probably can't speak more than that, but it really looks like the kind of thing that if we find safety, we should think about this for younger children with very red skin, which is why I'm excited about doing a ustekinumab study. Starting a child under 2 with some kind of an oral retinoid, pros and cons. Well, I think it depends on the severity, and I will tell you that I usually wait a few years to do that, but if somebody's very severe and the topicals, which of course would be safer, are not doing the job and it's causing issues, it is something to think about even in somebody who's young. But we often don't start that young for lamellar ichthyosis. We'll go with topical medications. If we can, we'll use a lot of moisturizers and just try to keep the skin in good shape. Now, why do we not do that? It's all about safety. We know long-term retinoids can lead to problems. One needs to be regularly checking blood for lipid levels and possibly even kidney, although I have never in my life seen a kidney problem with it. And then over time, of course, there can be issues with bones and joints and things like that we need to keep an eye on. And there are rare other associations. So we're careful about that type of thing. I think it's a matter of choice. When someone is very young, the impact of having a highly visible disease really starts coming maybe when you're 3, 4, start interfacing with other kids. So I don't think that's a high risk. It's really more of an issue for parents. And I think parents just need to learn to deal with this, and we can provide help with that. So I don't usually start in the very young ones myself. I usually wait until the child can join in some conversation and try to start it early enough if we feel that the topicals aren't doing it, to try to avoid some of the psychosocial issues, but we do it with eyes open and we have to be very careful. I will tell you there are 2 oral retinoids out there. Now, one of them is cleared very quickly. That's the one that's out there for acne. It's called isotretinoin. It's out of the body in a day or 2, and we even tell pregnant women they can get pregnant in a month if they're using that. So that would be the one that we'd favor, as opposed to something called acitretin, which can take 2 to 3, it takes months to clear fully, and we tell people not to get pregnant for 2 to 3 years. Now, if you use it when you're not at risk for getting pregnant, you don't have problems later on with fertility or risk of changes in babies. I want to make that clear. But nevertheless, the big problem with using the one that's out there for acne is that there's a very complicated program that's called iPledge that you have to be part of, even if you're 2 or 3 or 6 and not really at risk for getting pregnant. Even if you're a boy, and there's no risk for getting pregnant for them. So it requires you to be, at the very least, just checking in every month and talking to the office and having a new fax go to the pharmacy. We can only do 1 month at a time. So it's a little more complicated. And for that reason, I've sometimes used the acitretin if I have no concerns about pregnancy. I was the investigator for a study many years ago that was just in cells and doing things, trying just to develop some keratin 10 specific gold nanoparticles. That's not off the table, but what I found was that it was much more complicated because we're not just suppressing something, we have to suppress at the level of a single DNA base, which is not so easy. And so what we decided was to sit back until that program proved itself. And that program continues to move forward. There have been phase 1, those early phase trials done for psoriasis with agents that just need to suppress the gene and the protein production altogether. And that's targeting the receptor for interleukin 17, in fact, for one of them. So that could be an interesting way to treat ichthyosis perhaps in the future too, something I'm keeping in mind. That study stopped because it was clear that it was going to be extremely challenging, and we will think about starting that up again down the road in a few years if those move forward and the phase 2 trials look good. But otherwise, I think it's just too difficult until we have proof of the effectiveness of the technology. Yes, TALEN-mediated gene editing, you know, as it was, I went much too long. That's so early on that I didn't talk about it, but TALENs have gone the way of something called CRISPR-Cas, which is even better than TALENs. And what these do is they're able to recognize specific areas in genes, introduce the correct sequence after cutting out the bad sequence. So one of the problems with some of the dominant disorders, meaning passed on from parent to child, in which you have one of your 2 gene copies that has the genetic change, and it's enough to make the protein and have that protein mess up the works, is that you can't just add more of it. That probably would help to some extent, because you may shift the ratio of good to bad protein and gene. But even better would be to take the one that has the abnormal gene, take that certain segment of the gene, cut it out and replace it. And doing that sort of thing could be a permanent replacement. And you can do that with this technology called CRISPR-Cas. The problem is it's incredibly inefficient. So you would have to do it and sort out those skin cells in which you successfully did it, get it so it was all of those skin cells, make sure it was in ones that are going to just grow and grow and grow, the so-called stem cells, make those into sheets, and then put those on. Now, I will tell you that skin grafting is a way to go. You know, it's not perfect. You're going to have some scars from that. You're going to have some changes from that. It's something that we want to do for very serious conditions. If we can do it topically, it's just so much easier from a conceptual standpoint. And I hope someday we're able to just rub on something with CRISPR-Cas or something that suppresses the gene and changes the gene for solving some of these types where we have to get rid of the bad gene component, not just add something. And I'll say a little something about the gene therapy approaches that I was just talking about. So we'll probably get to this later. I saw there was more of a question. So the gene therapy that I talked about with topical delivery is just like topically applying anything that we have right now, okay? It is not permanently fixing it, but it approaches the problem in a highly specific manner that should be much more effective and very safe because you're just putting it on the skin. But the type of setup that's used, that viral vector, is not integrating. It doesn't get into the gene. So that brings with it a lot of good safety, but it also means that this is the type of thing that every week you would be putting on to keep it under control. It's not a permanent cure. Now, if you're grafted everywhere and it persists, and that's a big if, because that means you have to get it into the stem cells. And if it doesn't go into the cells that continue to replace the skin cells as they go to the surface and are shed, then you're just going to have it for a temporary period of time. And that's been some of the difficulty in some of the very early work that's been done so far with that, is that it's not a persistent change. It's not getting into enough of those stem cells. So just so you understand a little bit more, we don't have a cure yet, even with the gene therapy, because it has to be something that's persistent. And all of these grafts, unless you have it in the right cells that are not going to just get replaced and shed, you're not going to see permanent change. Wondering, okay, will the itch data help stop insurance refusal? As an example, could not get Tazarac covered. Yeah, you know, one of the problems is that insurance is sometimes helpful, and insurance sometimes comes with any excuse they can. And unfortunately for ichthyosis, everything is what we call off-label. There is nothing that has as its indication specifically tested for ichthyosis or a certain type of ichthyosis. Now, some of these companies will be coming out with that because they're doing all the testing right now. But things that we're using, like Tazarac, was all tested for psoriasis. The company that makes it put it out there with testing that allowed the FDA to approve it for psoriasis. There's nothing in the label about using it for ichthyosis. And that is an excuse then for insurance not to cover it if it doesn't want to. Having itch data in itself wouldn't make that happen. It would either have to be insurance being more generous in covering off-label indications, which sometimes your doctor can get by just pushing and pushing and showing how miserable kids are and showing what the harlequin ichthyosis looks like and showing that nothing else has worked. We sometimes can get this covered, but it takes effort. It takes letters. It takes potent letters that talk about the quality of life and the incredible burden of disease. Or the company could say, hey, we're going to do a study now and show that Tazorac works and get it on label and make it easier. But with the limited numbers with ichthyosis, a lot of companies won't do that unless they have something more specific. And we're delighted that companies are now doing this specifically for retinoids and other agents for ichthyosis. Let's see. We now refer to harlequin as CIE. Yes, it is a form of CIE. It's the most severe form of CIE. And in fact, the genetic changes on both of the genes that cause harlequin ichthyosis involve different areas of the same gene as causes a much milder form, which is classically called congenital ichthyosiform erythroderma. So it is part of congenital ichthyosiform erythroderma. Okay, I understand this trial using an engineered herpes simplex vector. Can you describe the details on the ability to administer more than once? Have there been studies confirming that it's not integrating? Yes, as I said, this is a special kind of non-integrating vector that happens to be built on herpes simplex virus. Are there concerns about dilution of the transgene, which would require multiple administration? So yeah, and I think I covered that too. We're putting it on once a week in the studies. We'll see how that goes. But this is not permanent. This is not integrating. It's requiring administrations regularly, and there's no evidence of antibodies to the engineered vectors. So everybody in these studies is getting baseline studies to see if they have antibodies against the herpes simplex, and so many people do, of course, because it's such a common virus infection. And so if they do not, we're making sure that they don't develop. If they do, we're making sure that they don't go up. Gene therapy would occur before bone marrow transplant. Ooh. Well, we're not doing bone marrow transplant for any of these forms of ichthyosis. There is something that's called stem cell transplantation. It's been used as gene therapy for many forms, but for something that primarily is on the skin, if we can do something that's on the skin, whether it's a transplant or whether it's a topical, it's far preferred to trying to introduce it into the bone marrow, because we don't know that anything we put in the bone marrow is even going to get to skin. This has been tried, for example, for that blistering disorder, epidermolysis bullosa, that I showed you before. That is a disorder where in many children, there's a tremendous amount of internal manifestations. So it's not enough for some of them to put it on the skin. It actually was for the boy I showed you in Europe. But for a while, they were trying to do bone marrow transplants, stem cell transplants. Honestly, didn't work very well, even though with wounding, that is a signal to get cells from the bone marrow to the skin. We don't have any evidence of that really being the case for these forms of ichthyosis. So I wouldn't even be thinking about bone marrow transplant here, not to mention there are lots of risks for it.
Dr. Peller,
Yeah.
this is Shannon Hamill. I'm the one who presented that question, and I'm just finding that with harlequin ichthyosis and my daughter's presentation specifically, it seems to be more systemic. I mean, she has cardio and liver, and I know some of our specialists have consulted with you on how to.
I think this is all related to the ichthyosis, because liver problems, cardio problems, as I told you, we see those kinds of things in psoriasis. And that's why she really should be on one of these agents as soon as possible.
They're considering starting her, I mean, with COVID, we were going to start her on Taltz with the knowledge that we'd probably be seeing you in June, which isn't going to happen. So we met with our rheumatologist on Monday virtually, and I guess we're going to start her on Taltz in early June and wean her off her prednisone right now. Is that what you think should happen?
Yeah, I have several people on it for psoriasis, or even the baby I mentioned to you with the SAM syndrome, and they've been fine. I mean, we're all sheltered in. There is a risk with infections in general with some of these biologics. It's not a big risk. I don't know of any increased risk with viruses. We know nothing about this SARS-CoV-2 virus, but so far it's not a big problem that we've seen. We're tracking adults with psoriasis very carefully. I'm happy to talk with you more about it offline. You can just email me.
That'd be great.
I'm happy to see pictures. I'm even happy to do a telederm clinical visit with you, official one, if you want.
That'd be
we
perfect.
can talk about that. But I think it's the right thing to try,
Okay.
and is the right medicine to try.
Okay, great. Thank you so much.
Amy, I want to be respectful of your time too. I know there's a couple more questions listed here. I don't know if you want to go through them or if you're
Just a few, so I will just quickly do that. We answered Sam's question. I think we answered Stefano's integrating gene editing strategy. So ultimately, to cure, you want gene integration, but you need to be able to direct that integration. And so CRISPR-Cas, which I talked about, can cut out and replace. Boy, I hope that goes someplace with topical, ideally. So over time, as you topically applied, you could actually change the gene and create something permanent and not have to just go through grafting. But I'm hoping so. You know, nothing happens overnight. We're going to be in a different place in 10 years. We're going to be in a different place in 20 years, things that we've never even thought of. And then I think the last question that I have is if the disease is detected before birth, is there a possibility of getting it? What is the scope of these gene therapies at the germline level? Well, that's pretty far off. There's gene therapy that's being done very, very limited in utero. It is largely being done, for example, for immune issues, where you can repopulate the bone marrow and blood, and then have that make a difference for the patients. I think it's a little hard to imagine how you're going to do that for the skin. Topical delivery is not going to be an option in utero with all the fluid around it. We can't do that. We could potentially do gene therapy very early on. But remember, anybody who's detected prenatally already has a lot of their skin being developed. This would be something more challenging, but possibly in the future. We can do gene editing if you know early on, and there are techniques now where if, let's say you have somebody in the family who already has ichthyosis and you decide you just don't want to have another child who has that, you can have a technique done called pre-implantation diagnosis, where you go through in vitro fertilization. You take any of the fertilized eggs and you look very early on at a stage where there are just a few cells that have divided. It's called the blastomere phase. You can take out a single cell, you can extract the DNA, you can analyze it, and you can just implant those that have the normal, at least one of the genes being normal if you need two for a problem, or having no gene with a problem if you have something that is just on one side and causes ichthyosis. But that's when you know somebody else in the family is affected, and that's really a preventive type of thing. We're a long way off from this, but gosh, I hope it's available if you know prenatally, and we're going to know prenatally because we're going to be able to do these big data arrays. You're going to be able to look at every gene. We're going to be sometime forced with having to deal with having lots of changes in our genes and then having to figure out how to interpret it and what's real or what's not, and potentially making decisions based on what we find. So it's going to be a whole new world, possibly in even just a decade. And I will warn you that it's not so simple looking at these genes, because we all have so many genetic changes, most of which, almost all of which, mean nothing and do nothing. We don't even know about them. And with that, I'll stop, Chris.
Okay. Oops, getting there. Hold on. Hold on. Oh, brother. I'm sorry. I was on the other screen as you were saying that. We're going to wrap up, and we wanted to thank you, Amy, very much for your time. You were a brilliant person for ichthyosis, and we're so glad to have you. We've had you for so long and on the Medical and Scientific Advisory Board. You are so well respected, and we really thank you for your time. I just wanted to mention to everybody, this has been Facebook Live tonight. So tomorrow, because there was a lot of information, and I know it's almost overwhelming, but tomorrow you can look at it and in the leisure of your own home and just go over the parts that you'd be interested in. I also wanted to just mention that we have several more talks for the end of the month, finishing out Ichthyosis Awareness Month, and it's all on our events site on our website. And then we hope we'll see you at the last day of the month to wrap up Ichthyosis Awareness Month with another Release the Butterfly concert. It was beautiful, beautiful concert, and we hope you will join us. So thanks again, and we hope to see you at the next one. Thanks, Amy.
Thank you. My pleasure. Bye, everybody.
Thank you,
Bye-bye.
Dr. Power.
Thank you, Amy.